For hemoglobin to transition between the R and the T state when it's supposed to, hemoglobin needs a little help from its surroundings. Here we're going to learn about some of the molecules that help hemoglobin move between states. An allosteric protein is one that has multiple subunits. When one subunit undergoes a conformational change, it is communicated to the other subunits. Based off our previous discussion of hemoglobin, you could probably guess that hemoglobin is in fact an allosteric protein. They're molecules that bind to hemoglobin and either activate or inhibit its binding of oxygen. We have a fancy name for these molecules called modulators. Oxygen itself actually is a modulator because it affects the binding of subsequent oxygen molecules. Modulators can either be homotropic, which means they are the substrate of the molecule, or heterotropic, which means the molecule is not the substrate of the protein. Oxygen is an example of a homotropic modulator, and during the rest of this presentation, we'll see some examples of heterotropic modulators. Remember when we talked about biological buffers and we mentioned that the bicarbonate buffering system was responsible for maintaining blood pH? Well, since hemoglobin hangs out in the blood, it's buffered by the bicarbonate system, and it turns out that this buffer system actually modulates hemoglobin's binding of oxygen. But let's back up before we get there. Let's recall that CO2 gets in the blood because it is a byproduct of glucose oxidation in the mitochondria. So for every glucose molecule consumed, 6 CO2 molecules are produced and released into the bloodstream. There are 3 ways for CO2 to leave the tissues. The first is that it dissolves in the aqueous cytoplasm, it becomes converted into bicarbonate, or it can hitch a ride on hemoglobin by becoming a carbamate adduct. For the first 2, when oxygen is dissolved in water, it turns into carbonic acid. If you're wondering why increased CO2 emissions is bad for the ocean, this is one result. Dissolved CO2 makes the seawater more acidic. The carbonic acid is a buffer that then exists in an equilibrium between itself and bicarbonate and a proton. This buffering system between carbonic acid and bicarbonate is what maintains the blood pH. We talked previously about how to maintain the physiological pH of 7.4. The ratio of bicarbonate to carbonic acid is 20:1. Although this is out of the typical range of a buffer, the blood can make this happen because the body is an open system. How is it an open system? Well, CO2 is constantly coming into the cells and exiting the lungs through respiration. The enzyme that makes this happen is carbonic anhydrase, which has the ability to convert between CO2 and carbonic acid. It's what turns CO2 in the tissues into carbonic acid, and now it turns bicarbonate into CO2 in the lungs to allow it to be breathed out. I mentioned a bit ago that this buffer system directly modulates the T to R transition. How is this? Well, one way is through the Bohr effect. In the tissues where the mitochondria are oxidizing fuel, which produces CO2 that is eventually turned into bicarbonate and a proton, this is the proton that protonates histidine 146. When the proton concentration is increased as a result of CO2 production, there are more protons around that can decrease the pH and then protonate histidine 146. You'll recall that the pK of histidine is very close to physiological pH, and this is where it comes into play. Because when histidine 146 is protonated, it can form a salt bridge with aspartate 94. This new salt bridge stabilizes the T state and by extension destabilizes the R state. As a result, hemoglobin favors the T state and releases oxygen into the tissues. This makes sense based on hemoglobin's role as an oxygen transporter. Therefore, we can make the conclusion that at a lower pH and a higher concentration of carbon dioxide leads to a stabilization of the T state, a weaker oxygen affinity, and a release of oxygen into the tissues. We can see this in the graph at the right, where at a lower pH, the P50 shifts to a higher PO2, which means it takes more oxygen to half saturate hemoglobin, indicating a lower affinity and a favoring of oxygen release. The other way in which the bicarbonate buffer system modulates hemoglobin is through the formation of carbamate adducts. In this instance, CO2 literally acts as a molecular hitchhiker on hemoglobin. What happens in the tissues where there is a high concentration of CO2, CO2 binds to the N-terminus of hemoglobin monomers, forming a carbamate adduct. It turns out that the carbamated hemoglobin is less stable when bound to oxygen. Therefore, the carbamylation favors the T state and the release of oxygen. The protonation of histamine 146 that results from the decreased pH that comes from increased CO2 production and carbamylation lead to an inverse relationship between CO2 concentration and oxygen affinity. Another way that the body regulates the transition between the T and R states is through the production of 2,3-bisphosphondiglycerate, or BPG for short. BPG is a heterotropic allosteric modulator of oxygen binding. What it does is stabilize the T form of hemoglobin. If we look at the black line on the graph, which is the oxygen binding of hemoglobin without any BPG present, we can see that hemoglobin, again, looks a lot like myoglobin. This is because without any BPG in the bloodstream, hemoglobin would always be in the R state. This is a problem because it would mean that hemoglobin would want to hold on to oxygen, not release it, and our tissues wouldn't get the oxygen they need. Essentially, you would just have another myoglobin. However, our bodies naturally produce BPG. You'll notice that for the blue line, which represents the concentration of BPG in the bloodstream that would happen in a normal human at sea level, that the curve regains its sigmoidal shape and clearly has a lower affinity for oxygen at the level in the tissues and a higher affinity for oxygen at the level of oxygen that will be in the lungs. There's also a green curve, which represents the binding of hemoglobin to oxygen at high altitudes. At high altitudes, less oxygen is available. This means our body won't be able to bind as much oxygen as it could at sea level. Anyone who's tried to do any sort of athletic activity in the high mountains know this is the case. If the body can't bind as much oxygen, then the tissues won't be able to get released as much as they need either. Therefore, at high altitudes, the body compensates for this by producing slightly more BPG, shifting the curve and the P50 to the right, which encourages more hemoglobin to adopt the T state and release oxygen, even if the oxygen concentration is lower. Since BPG is a heterotropic modulator, you may be wondering where it binds. If hemoglobin were a donut, BPG would be the donut hole. It binds right to the center of hemoglobin, locking it into the T state. The last modulator we'll talk about is carbon monoxide, or CO. If you're wondering why some people have carbon monoxide detectors in their house, I know I did when I was a kid, this is why. Let's say that you have a person who is anemic, or they don't produce enough iron to make the heme to sit in each hemoglobin monomer. These people typically have hemoglobin molecules with half of the oxygen binding capacity of normal hemoglobin. But their hemoglobin still has allostery. This means that their hemoglobin can bind oxygen in the lungs and release it in the tissues because it can transition between the T and the R state. It just binds and releases half as much oxygen. Let's say you have carbon monoxide bound to 50% of your hemoglobin subunits. Not so much. It turns out that carbon monoxide binds to hemoglobin in the exact same spot that oxygen does, except carbon monoxide is a much better ligand than oxygen. The binding is tighter, in fact, 200 times more tighter than oxygen. So what happens is carbon monoxide effectively blocks O2 from binding and locks hemoglobin in the R state. This causes the other subunits to be locked into the R state as well, and cooperativity is lost, demonstrated by the hyperbolic curve. So people with carbon monoxide poisoning are doubly screwed. They can only bind half the oxygen in a normal person, and then they can't even release it, so the tissues become deprived of oxygen. Unless that person is taken immediately to the hospital to undergo hyperbaric oxygen treatment, they will die. Let's take a quick minute to summarize how these different modulators affect the T and the R states. This is the way I like to represent the relationship, but you're encouraged to come up with your own if that helps. So higher oxygen concentrations favor the formation of the R state, whose function is to bind oxygen. This makes sense, because in the lungs, where the O2 concentration is high, hemoglobin wants to bind oxygen. The competitive binding of carbon monoxide to hemoglobin also forces the R state. Conditions that favor the T state as opposed to the R state is a lower pH, which results in a higher proton concentration, adiprotonation and formation of that salt bridge that promotes the T state, higher carbon dioxide, which again leads to a higher proton concentration or carbamylation of the hemoglobin monomers, and also higher concentrations of BPG. This makes sense because the pH is lower and the CO2 concentration is higher in the tissues where oxygen needs to be released, which happens in the T state. Hemoglobin is really important, and when it gets messed up, bad things happen. We can also see these defects in hemoglobin structure as a demonstration of the sequence structure-function relationship. So there's a glutamate 6, on the hemoglobin alpha-beta interface that has a tendency to be mutated to a valine. This happens when there is an alteration to the DNA sequence that leads to a different amino acid, specifically a valine instead of a glutamate. Now, this has to happen for some kind of a reason. So the theory is that people who have one copy of a glutamate to valine mutation carries a protective advantage against malaria. But again, only if the people have one copy, so that mutation's only on one chromosome. People who have two copies of hemoglobin with the glutamate to valine mutation develop sickle cell disease. So what happens is if you replace a negative charge on that alpha-beta interface with a hydrophobic amino acid, the valine will form a hydrophobic interaction with hydrophobic amino acids on the other side of the interface. If enough mutant hemoglobin molecules form those interactions, they can form strands which will eventually crystallize and come out of solution. This is analogous to those proteins in diseases such as Alzheimer's that also slightly unfold, glom together, and then can form fibers. Those crystallized fibers will block the capillaries, which leads to pain and organ malfunction, as well as a lower concentration of functional hemoglobin. Red blood cells with this defective hemoglobin often adopt a sickle shape, leading to the moniker sickle cell disease.